Hey folks, I hope that you're hearing this okay. I don't know what time you're watching this, so I'm not going to say good morning or good evening, but hello. So we have got a basic lesson here. Obviously, there are things I can't do in this format that we could do in class. I don't have a whiteboard. I'm doing this from my house, so I don't really have a mechanism of doing drawings for you or anything. So I might recommend that when you are listening to this, you might want to follow along in your textbook. In particular, look at the sections in Chapter 4 where you're going to be seeing diagrams of prokaryotic and eukaryotic cells. That will give you a better look at the structures that I'm going to be talking about so that you have some idea what is going on here, and also so that you can see the terminology that I'm going to be using. I'm not going to be writing things on a board clearly, so refer to the textbook and that will give you a way to get the terms. I'll try to spell things here as I bring up terms that might or might not be familiar to you. Now, we're not quite done talking about prokaryotes yet, but the main thing we left off with concerning them, we just talked about antibiotic-resistant bacteria, this problem of superbugs, this issue with the fact that we are putting evolutionary pressure on bacterial populations. We're putting them in a situation where they are forced to adapt to an environment full of antibiotics in order to survive, and as a result, they're building defenses against the primary weapons we use against them. Now, like I said, it's important to remember the antibiotic is not causing the bacteria to become more powerful. The antibiotic is not inducing mutations in the bacterial population. Rather, what the antibiotic is doing is creating an environment in which only the most resistant bacteria are going to survive and thrive. And so it's drawing upon existing variation in the population. Bacteria mutate very easily. Their readily damaged DNA, because it doesn't have the protections that your DNA has, is very subject to environmental forces that can change it in some fashion. Most of those changes aren't good, but when the bacterium has a helpful change in an environment full of antibiotics that is challenging them, the bacteria that are most resistant, that have the variations that make them more likely to get by, are simply going to do better. And the more we overuse these tools at our disposal, these antimicrobials, these antibiotics, and so forth, the more rapid the process of bacterial evolution. This is not to say that antibiotics don't have a place. They are used for a reason. They can prevent serious infections. They can kill people. Likewise, antiseptics certainly have their place. And if you're in an environment that's very high risk of infection and there's no means of washing your hands, the hand sanitizer is helpful. But the point is to treat these things as special occasion tools, not as go-to all-the-time tools. Ordinary soap and water with no antimicrobial properties is perfectly fine for ordinary hand washing and countertop cleaning and so forth. Likewise, antibiotics, get them only when you need them, which is not when you have a viral infection like a cold or a flu. That's not a bacterium. It's not alive. It can't help you. Likewise, hold off on getting it until you need it. Don't use it if you don't have to. And when you are using it, make sure that you actually use the whole course of the antibiotic. Go all the way through with it. Don't throw away the excess. Even if you start feeling better, finish out the course that your doctor prescribed you. That's finishing off the stragglers. That's taking out the bacteria that might have been left behind at the end. All right? Now, we don't want to spend too terribly much time on that. Take a microbiology class. You'll hear a lot more about antibiotic-resistant bacteria and their defenses and so forth. I've given you the fundamentals here, but you can get more out of it out of another course like that. We have a good micro class here at CCAC. And I can chat more with you about it on your own if you're thus inclined. But one last thing I wanted to talk about concerning microbes and prokaryotes in particular, concerning prokaryotic cells, is moving around. How do they move? How do they get from point A to point B? This is a thing we call motility, M-O-T-I-L-I-T-Y, motility, the ability of an organism to move. Now, when you're a single-celled organism, clearly you don't have limbs, you don't have muscles, you don't have any of those multicellular structures that enable you and I to move around. So how do you get from point A to point B if you're a prokaryote? Well, the answer for a lot of them is you don't. About 50% of all documented prokaryotic species have no mechanism for motility. They don't have any structure or feature that is going to help them to move under their own power. They can't do it. And as a consequence of that, they're basically just carried around with environmental forces. A non-motile prokaryote is carried by water currents, it's carried by air currents, it's carried on the body of an organism that moves them from point A to point B. They have no power to change their own position. But there are motile prokaryotic organisms, and there are two primary structures that they use for motility. Now, if you look at a microbe under the microscope, you look at a bacterium, for example, under the microscope, you might find that it has lots of little hair-like fibers extending out from the cell wall all around its surface. Those structures are called cilia, C-I-L-I-A, that's the plural form. One of them, if you're talking about one of them just by itself, is a cilium, C-E-I-L-I-U-M. Sometimes they're called pili, P-I-L-I, singular form pilus, P-I-L-U-S, if you're talking about just one. The cilia or pili fulfill a couple of functions. They're little tiny hair-like fibers. The word cilium from Latin means hair. Pilus is Greek. That also means hair. But they're not actually hairs. What they are fibers of the motion protein actin. And they fulfill a couple roles for the organism. For starters, they can grip onto surfaces. They can help the organism to stay in one place if it wants to, or also help it to grip onto other nearby cells. Colonial bacteria that live in groups very often cling to one another via their pili. Another thing that these structures can do is that they kind of weave back and forth. See them like the oars on a boat, kind of waving around in their environments, pulling the organism through the medium that it lives in. So the cilia act like oars on the bacterial ship to carry it around in the environment, get it from point A to point B. Now, there's another kind of motility structure that you'll find in prokaryotes, and that is called a flagellum. F-L-A-G-E-L-L-U-M, flagellum. If you're talking about multiple ones, the plural is flagella, F-L-A-G-E-L-L-A. Now, the flagellum, rather than being little short hair-like fibers, is a long whip-like fiber found at one end of the organism. The term flagellum from Latin means whip. Sometimes you'll find just one flagellum. Sometimes you'll find a cluster of flagella all together at one end of the organism. Think of the flagella as being kind of like the propeller on a boat, that it whips back and forth, driving the organism through its environments, generating a force to propel it forward. Now, your flagella are going to be a thing that you don't usually have both. Some organisms do have both. Some have cilia and flagella. Some have one or the other. Now, the cool thing about these structures is that although they originated in bacterial populations, they've been repurposed for use in more complex multicellular life. Eukaryotes can have cilia and flagella as well. But in your body, they do something very different. The flagellum, that whip-like fiber at the end of the cell, in our bodies, I think you all know where this is found, sperm cells. The male reproductive cell, the spermatozoa or sperm, has the flagellum at one end, whips around to propel it through the environment. The sperm is one of the very few cells made by the human body that has to be able to move under its own power once it exits the body and is searching for the egg. So the flagellum gives it that propulsive ability. Now, cilia, you find those in your body in a few places, but not in cells that need to move around. In us, the cilium has been adapted to do a new job in a cell that's fixed in one place. You will find ciliated cells lining your respiratory tract and your nasal passages, your throats, down into your lungs, where those cilia weave back and forth and push mucus over their surface. They carry mucus coats out of the lungs, out of the lower respiratory tract, out of the upper respiratory tract, push all that mucus down into your throat where you reflexively swallow it. You don't even notice you're doing it, but you do this all the time. Having that reflexive swallowing action clears out that mucus from your respiratory tract, keeps you breathing efficiently, and also clears out all the particles that are captured by that mucus. When you breathe in, you're inhaling all of the pollutants and particles in the air around you, whether that's air pollution, dust, bacteria that are airborne, all sorts of things. Those things get trapped in the mucus coat inside your nose and inside your throat and inside your lungs. Pushing that mucus coat up and out is a good way of clearing things out. That's what those cilia are good at. They don't help the cells move around. The cells lining your respiratory tract don't go anywhere, but the cilia on their surface still beat and push, and that shoves the mucus coats up and out. One of the persistent dangers of long-term smoking is that if you're a smoker, you're going to find yourself losing those cilia over time. We all lose cilia as we age. We all experience a loss of cilia as we get older. One of the consequences of this is that as you get older, you have a harder time clearing mucus out of your respiratory tract. And that means that you're going to be more prone to respiratory infections. You all know older people are more prone to pneumonia, bronchitis, things like that. We all experience a little bit of ciliary loss because we're all inhaling environmental pollutants that damage those sensitive cilia. But if you're a smoker, or if you're working in an industry where there's a lot of smoke, a lot of pollutants in the air, and you're not wearing protective breathing equipment like coal miners used to do, they get black lung disease, right? What's happening here is that those pollutants are destroying the cilia that line the respiratory tract. And that makes it harder for you to eliminate those mucus coats that build up. And you have to develop a forceful cough as a way of propelling them out. We call it the smoker's cough. You know, you all know a smoker who does that. It's because they've lost a lot of their cilia that are helping them to do that job. We all experience ciliary loss as we age, but if you smoke or around a lot of heavy pollutants, you're going to experience it more, worse, and younger. They'll grow back a little over time. You stop smoking today, you'll definitely regenerate them, but the older you get and the longer you've been a smoker, the harder that becomes to do. And I'm here to tell you folks, I know a lot of you smoke. You're going to shrug and say you got to die of something eventually. I've watched people die of lung cancer and neck and throat cancer. I've had 7 relatives die that way. It is a horrible, choking, gurgling, painful, lingering, slow, terrible way to die. You don't want to die that way. Save your cilia. Save a whole lot of money. What you spend on cigarettes in a year, you can use to take a trip to Disney World. Everybody likes Disney World. So we've done this thing where we've repurposed the cilia in our body, not for a structure of motion, but to help out with a different job. We also have them in our intestines. In your intestines, they actually increase the surface area of the intestine to give you more surface area for absorbing nutrients. You have little finger-like fibers in your intestines called villi that extend into the interior of the intestine. They are covered in ciliary fibers called microvilli, and all those lots of little fibers greatly increase the total internal surface area of the intestine over what it would be if it was a smooth tube. With the help of all the cilia and the villi, the intestines have a surface area 300 times greater than they would have on their own. Those cilia help to push food through the intestines, but they also provide more space to absorb the nutrients from that food, which means your body is getting more out of it that way. There's a name for this, by the way, in evolutionary science. When a structure that evolved for one purpose is repurposed by another organism to do another job, like the way cilia started for motion and have been repurposed in your body to move other things around within your body rather than moving cells, that's called exaptation. E-X-A-P-T-A-T-I-O-N. Exaptation. It's the evolutionary repurposing of an old structure for a new function. All right? So this is all I really want to say to you about prokaryotes for now. We've already kind of started segueing away from prokaryotes into eukaryotes by talking about some of the things cilia and flagella are used for. They're not strictly prokaryotic structures. They're found in a lot of single-celled eukaryotes, and as I've indicated, they're found in a lot of structures in your multicellular body as well. But now it's our time to focus on the eukaryote a bit. We're going to mention prokaryotes again in this class. They're going to come up on more than one occasion. I'll mention where they're relevant and how they differ from eukaryotes when we talk about metabolism and other things. But in general, we're kind of setting them aside for now. We're going to move on to the kind of cell that's going to get most of our attention in this class, the eukaryotic cells. Those are the cells that we have. They are the cells that make up every animal, every plant, every fungus, every protist. 4 of the 6 kingdoms of life, the kingdom Animalia, the kingdom Plantae, the kingdom Fungi, and the kingdom Protista, the single-celled eukaryotes. All of those are made of eukaryotic cells. Eukaryotes have been around for about 2.5 billion years, billion with a B, so they came a good bit after prokaryotes. Prokaryotes had already been around for 1.5 billion years, before the first eukaryotes came on the scene. But the eukaryotes have done pretty well for themselves. Obviously, we've developed several multicellular kingdoms of life within the domain eukarya, including the one that we ourselves belong to. Pardon me. So what makes a eukaryote special? What makes them different than a prokaryote? And again, I recommend, since you don't have a diagram that I'm drawing to look at, go into your textbook, find in your chapters on the cells You'll find some nice diagrams of eukaryotic plant and animal cells there that you can check out. You might want to grab one of those diagrams, look at that while you're following along here with my discussion, so you have some idea what's going on and you can see these structures. It's probably better than my drawing anyway. Now, what makes a eukaryote special and distinct? A couple of things. First and foremost, the obvious, distinctive, defining feature of being a eukaryote is that you have a nucleus. N-U-C-L-E-U-S, nucleus. That is the structure inside of the cell that encloses and protects the DNA. The nucleus is a protective chamber for the DNA. Most cells have a single nucleus. A couple of kinds of cells, like muscle fibers, have multiple nuclei. The nucleus is usually the biggest thing in the cell. Sometimes it takes up as much as a quarter of the available space. It's usually found somewhere close to the center of the cell. There are some exceptions where the nucleus is found in other places, but that's the norm, to have them near the middle. Now, the nucleus is usually a roughly spherical organelle. In some cells, they're kind of more sausage-shaped, like in muscle cells, they're more elongated, but they're usually spherical in configuration. The nucleus is surrounded by its own membrane, a double phospholipid bilayer. Imagine you have two phospholipid bilayers, stack them up back to back. You're going to have the membrane surrounding your nucleus. That membrane is called the nuclear envelope. And the nuclear envelope encloses and protects the DNA and the rest of the contents of the nucleus. Being a double phospholipid bilayer gives it a considerable amount of safety. That's good protection for the DNA. Anything that has to harm that DNA not only has to get through the external membrane and possibly the external cell wall if the cell has one, but then it has to get through that nuclear envelope as well. So it's a good protective layer. There are openings in the nuclear envelope. They're called nuclear pores, P-O-R-E-S. A pore is a hole or opening. Nuclear pores are going to provide access to the nucleus. Not a lot of things can get through them. Mostly nutrients get in and out, nucleotides get in and out, and RNA molecules get in and out. Not much else goes in and out of the nucleus. And unfortunately, certain viruses are able to trick the nucleus and slip through the same openings as RNA molecules and get in there and attack the DNA directly. Herpes viruses are notable for doing that. They hide themselves inside the nuclei of cells, which makes them really difficult to get rid of once you're infected. And it's why herpes keeps coming back again and again, and you keep getting repeated episodes of it once you've had it. Within the nucleus, you'll find a little pocket of cytoplasm that's separate from the rest of the cytoplasm of the cell. Sometimes it's called the nucleoplasm, N-U-C-L-E-O-P-L-A-S-M, nucleoplasm. You'll find the DNA in there, obviously. The DNA is enclosed within the nucleus. You might recall eukaryotic DNA is chromosomal. Eukaryotes have chromosomes, meaning their DNA is packaged into these tightly compressed units of DNA wrapped around proteins. We'll talk more about chromosomes later this course. Having chromosomal DNA means that you can fit more DNA into a smaller space. So my eukaryote, by having chromosomal DNA, can have a bigger genome. It can do more things. Your average bacterial genome is about 1,000 genes. Your average human genome is about 25,000 genes. We have more capabilities, and that chromosomal packaging system makes that possible. You wouldn't be able to maintain that big and stable of a genome without the protection of the nucleus. The DNA is not the only thing in there. You will also find a sort of dark-colored region when you stain the cell. There will be a dark staining region near the DNA called the nucleolus, N-U-C-L-E-O-L-U-S, nucleolus. And the nucleolus is a region near the DNA, which is the primary site of RNA synthesis. When the cell is making RNA, which is the messenger molecule for DNA, sending out instructions from the DNA, the nucleolus is where it's going to be produced. We're going to talk more about the nucleolus and that whole process of RNA manufacture later this semester. So you got your nuclear envelope around the outside, nuclear pores in that envelope to allow access to certain molecules, nucleoplasm inside of it. Floating around in there, you'll find your DNA in chromosomal form, and you'll find your nucleolus near the DNA. That's your nucleus for you. Kind of think of it as the command center of the cell. Now, Another big defining feature of being a eukaryote, a really important feature of being a eukaryote, is the fact that the nucleus is not the only specialized internal chamber. You will find that much of a eukaryotic cell, not all of it, but a good part of it, is compartmentalized. There are lots and lots of distinct membrane-bound compartments within the interior of the cell. There's a whole network of internal membranes within a eukaryotic cell that we call the endomembrane system, and that's endomembrane, endomembrane. Prefix endo at the beginning of a word, whenever you see that, means within. So it's with internal membranes, membranes within the cell. The endomembrane system is a direct outgrowth of the plasma membrane, or rather ingrowth, I should say. It's growing into the interior from the plasma membrane. It connects to the nuclear envelope, and portions of the endomembrane system compartmentalize the cell, divided up into a number of distinct inner compartments. We call those compartments organelles. And you guys know that word already, organelles. They are, you can think of them as like the organs of cells, specialized chambers that do particular things. Now, having the cell subdivided in this way confers several advantages. First and foremost, lots of specialized compartments, as I've said in here before, they can do particular things better. Just like you have a specialized workshop that has tools to do a particular kind of job, and a specialized reading room that you can go into in the library and so forth, the different organelles have structures that make them better at particular tasks. And that means that the cell can do those things better. It has more capabilities. Secondly, because the cell is compartmentalized, it has lots of distinct chambers. Remember how we said prokaryotes, everything that happens inside the prokaryotic cell has to happen in the same internal chamber. Well, eukaryotes are divided up. They can have lots of different chemical activities going on in different compartments at once. So not only can a eukaryotic cell do the things it's doing better, it can also do more things at once. A eukaryote can simultaneously maintain a larger range of activities at one time without the danger of them interfering with one another. And that means that your eukaryote can have a more sophisticated range of responses to its environment. It has more capabilities. Furthermore, the endomembrane system allows a degree of control over where things go inside the cell. You can close off or open up certain chambers, open up a channel to allow molecules to go one place or not, or close it up to keep them from going another place. And in that way, the cell can regulate its chemical contents a little bit better. So having an endomembrane system, having organelles, confers a lot of advantages on my eukaryotes. It can do more things. It can manage more complexity. It can do those things better. It can even, potentially in some species, manage the coordination of its activities with other cells. This is one of the keys to being multicellular, is having a cell that is complex enough that it can coordinate its activities with those of other nearby cells. So this is an important precursor step to being a multicellular form of life. Now, the endomembrane system does have some drawbacks. First and foremost amongst them is that we have an energy cost. All those structures are expensive. You have to do more things. Being a eukaryote means that you're going to spend way more energy to carry out your life activities. You've got more parts to maintain. You've got more activities to run. They all require energy. They all require raw materials. So being a eukaryote is more energetically and metabolically expensive than being a prokaryote. You've got more stuff to do. Secondly, the endomembrane system, with having those isolated organelles, one of the problems here you can encounter with this is that greater complexity is not always beneficial. Think about it. Which of these devices has more things that can go wrong with it? This device or this device? This one's got more parts. It can do more things, but more parts mean more things that can fail, more things that can go wrong. And a consequence of this is that a eukaryote with all those complex organelles, that can be a problem. If an organelle can get damaged, then potentially that organism is in trouble. It may have, that cell might have the inability to do a thing that it's supposed to be doing, or to do it as well as it's supposed to be doing it. And that means that that organism might, or that cell anyway, might begin to fail, might even die as a consequence of that. Especially if it's relying on a specialized structure to do a particular job, specialization can kill you. And therefore, that added complexity does come at a cost, not only a cost in materials and energy, but also the cost in the danger of having lots of sophisticated parts creates more points of entropy, more places where things can go wrong. So, benefits, drawbacks. But you'll find that on balance, the benefits of being a eukaryote significantly outweigh the drawbacks. That endomembrane system, those internal compartments, give it more capabilities. It can get larger, it can do more things, it can respond to the environment in a wider range of ways. And natural selection, by and large, has said this is a good solution. They wouldn't have lasted if it was not an effective evolutionary solution. So what I want to do now, we've kind of established the basics of what it means to be a eukaryote. We've established that you've got greater complexity, more parts, internal compartmentalization, specialized chambers that can do particular things. I want to talk for a bit about what you find in that eukaryote, what you're going to find in there, the distinct parts that it's going to have, some of the different specific organelles that you're going to encounter, and the jobs that they do. Now, I'm going to be naming here a number of important organelles. I'm not listing every possible organelle that the cell might have. I'm listing some of the better known organelles and structures that a lot of eukaryotes possess. Individual cell types might be missing some of these. Individual cell types might have other structures that others lack. But this is kind of, you might say, a generalized eukaryote, like the most common organelles and other structures that a typical eukaryote will contain. First off, let's talk about internal division of space. You have the organelles, those compartments within the cell that are carrying out specialized activities. Not everything in the cell is enclosed by organelles. The rest of the cell, the space that is enclosed by the plasma membrane but not enclosed by organelles, all the other space inside the cell is called the cytosol. C-Y-T-O-S-O-L, cytosol. Cytosol is basically, just the free space, if you will, inside of the cell. It's filled up with cytoplasm. A lot of chemical reactions happen in the cytoplasm. A lot of metabolism happens in the cytoplasm. So this is the site of a great many important chemical activities, and it's the site where a lot of things move around from place to place. So not everything in the cell is compartmentalized into organelles. We've got the nucleus, obviously. You're going to have ribosomes in there. Eukaryotic cell, just like a prokaryote, has a number of ribosomes in it that are going to be helping to manufacture various proteins. These are things you know already. Now, not everything in there is an organelle. An organelle is a compartment. An organelle is a structure that's got its own membrane around it. That's why a lot of textbooks call them membrane-bound organelles. So a ribosome isn't an organelle. A lot of times you'll see a list of organelles that ribosome will be included on and the cytoplasm will be included on. The cytosol isn't an organelle. It's not a membrane-bound. I guess technically it's a membrane-bound compartment because it's the space inside of the cell and the whole cell is membrane-bound. But when I say organelle, I'm specifically referring to a distinctly membrane-bound space inside of the cell. There are plenty of other structures in there that are important cellular structures, but they're not necessarily organelles. Now, before I start getting into particular organelles and what they do, I want to talk a little bit more about that endomembrane system, the membrane itself, the internal membrane of the cell. We've got sheets of membranes woven into the interior of the cell, separating it into compartments and channels and so forth. As I said, the endomembrane system makes up the walls of organelles, makes up the nuclear envelope. But the membrane itself has some important functions. There are regions of membrane within the cell, inside parts of it, that are doing some special activities of their own. We refer to those internal membranes as the endoplasmic reticulum. It's a bit of a mouthful there, E-N-D-O-P-L-A-S-M-I-C. Endoplasmic, that's the first word. Second word, reticulum. R-E-T-I-C-U-L-U-L. Reticulum. Endoplasmic reticulum. We often abbreviate it in biology as the ER, capital E, capital R. I'm okay with you using that shorthand. The endoplasmic reticulum is a general term for the internal membranes of the cell. In addition to forming divisions within the cell, the endoplasmic reticulum has some functions of its own. For starters, let's divide it up a little bit. There are two kinds of endoplasmic reticulum. We can talk about the rough endoplasmic reticulum or the smooth endoplasmic reticulum. Rough ER, smooth ER is how they're often referred to. Rough endoplasmic reticulum gets its name because if you look at it under the microscope, it appears to be a bumpy membrane. It looks like it's got little bumps all over its surface. The reason for that is because the rough ER is studded with ribosomes. Lots of ribosomes are embedded in the rough endoplasmic reticulum. And of course, you know what a ribosome is already. The ribosomes are the protein factories. They synthesize all the different proteins that cell requires. Now, not every ribosome is attached to the rough ER. Some of the ribosomes are freely floating in the cytosol. And there is a difference. The ribosomes that are floating in the cytosol, that are just out in the cytoplasm of the cell as a whole, they are mostly making proteins that are going to be used within the cell itself. Proteins that are going to stay inside that cell to carry out particular jobs. On the other hand, the ribosomes that are bound to the rough endoplasmic reticulum, their proteins are usually either being moved out of the cell, exported to somewhere else in the body, or they are being used as part of a membrane. They're being incorporated into a membrane to help that membrane do a particular thing. I told you guys that the plasma membrane of the cell has a lot of proteins built into it as part of its functions. Most of the time, those membrane-bound proteins are going to be built by ribosomes found on the rough endoplasmic reticulum. Not a hard and fast rule. There are certainly proteins made by the rough ER that are being exported. There are certainly proteins being made by the smooth ER that are going to be membrane-bound proteins. It's not a clean rule. But general rough ER, think that protein is going out of the cell or becoming part of its membrane. Smooth ER, think that protein is staying within the cell to help it carry out its own jobs. Now, if I look at the smooth endoplasmic reticulum, smooth ER gets its name because it looks nice and smooth under the microscope. It has very few ribosomes on it. Smooth endoplasmic reticulum has two big jobs. For starters, the smooth ER is a site for lipid synthesis. A lot of the lipid molecules that are manufactured by your cell are produced on the smooth ER. It's a working surface where those molecules are manufactured. Secondly, you're going to find that the smooth ER carries out a vital role in detoxification. A lot of enzymes that break down toxic molecules are going to be attached to the smooth endoplasmic reticulum. And so it plays a vital role in helping the cell to clean up toxic waste materials. And those toxic waste materials are sometimes things you take in from the environment. Other times, they're metabolic byproducts. They're things your body produces as a waste product from its own reactions that have to be broken down so they don't do you harm. You all have heard that thing that, we all supposedly have 5 pounds of toxins in our body or whatever. That's not true. We do not have piles of toxic sludge just lying around in your body. Your liver is pretty good at processing most of that stuff. Actually, you're going to find more smooth endoplasmic reticulum in the cells of the liver than in any other cells of the body. The liver cells have a much larger volume of smooth ER than other cells do. It's important to their detoxification role. Now, important thing about this. Let's just suppose your body is being challenged by a toxin on a regular basis. You're exposed to this toxin again and again. Your cells are going to say, uh-oh, we need to get better at processing this. And the way they're going to do that is they're going to add more smooth ER. They're going to increase their volume of smooth ER to increase their processing power so they get better at breaking that toxin down. One of the consequences of this is that after a while, you can build up a tolerance, as we put it, to a toxin. That if you're repeatedly exposed, it doesn't affect you as much. Somebody who's been a heavy drinker for many years will not get drunk as easily. They're going to need more alcohol to get the same buzz because their body has built up a tolerance. If you're a snake handler for a living, if you're one of those people whose job requires you to handle poisonous snakes on a regular basis, you're a herpetologist who studies snakes for a living, or you're a zookeeper, or you're one of the people who quote-unquote milks snakes for their venom to make anti-venom out of for snake bite treatments, then what a lot of professional snake handlers do is they will deliberately expose themselves to small concentrations of snake venom on a regular basis, little tiny diluted amounts of venom, because that helps them to build up a tolerance so that if they're actually bitten by a snake in the course of their work, it doesn't hit them as hard. Unfortunately, we can also build up a tolerance to helpful drugs. Sometimes if you're on a medication for a long period of time, then you can build up a tolerance to that med, and you'll need a higher dosage to get the same effect, or it might stop being effective for you altogether. This is especially a problem with pain medications. particularly with opioid drugs. If you've been on pain management long-term for any kind of reason, you'll find that if you're on the same pain management drug for a long time, your body will build a tolerance to it. And that's one reason why people on long-term pain management often switch their drugs periodically. It's a way of making sure that you don't build up a tolerance to something that you need too quickly. You can lose tolerance after a while, but in general, once our cells have learned that, they don't tend to give up that capability easily. And this is one of the big problems that leads to opioid addiction. You know, people take the opioid drug for pain management, they build up a tolerance, they need more of the drug to get the same effect, they increase their dosage, they build up more of a tolerance. It becomes a vicious cycle. Now, of course, the drug that humans expose themselves to on a regular basis more than any other single drug in large amounts is alcohol. And what this means is if you drink a lot on a regular basis, you're going to build up a tolerance. You're going to need more alcohol to get the same buzz. But as you guys know, somebody who is a long-term drinker runs some risk of some liver problems, right? Because what's happening is that as the liver becomes accustomed to a higher and higher amount of alcohol, the liver cells say, let's add more smooth ER to process that toxin. That means that you break down the alcohol faster, you don't get buzzed as quickly, you don't get drunk with the same amount of alcohol that would make somebody who was a less experienced drinker than you, if we can say experience here, drunk. You build up a tolerance. So you drink more alcohol to get the same effect, and this is one of the reasons why alcoholics will tend to increase their consumption over time, and eventually those liver cells begin to suffer. Because what happens is they build up so much smooth endoplasmic reticulum that it begins to choke out other functions of the cell. The cells begin to swell up and they begin to die. And that, of course, puts more strain on the rest of the liver to pick up the slack from those dying cells that are releasing toxins and aren't doing their job anymore. And that makes them more likely to die. And this is a progressive problem. We know this problem, of course, as cirrhosis. C-I-R-R-H-O-S-I-S. It's got a weird spelling, that silent H in the middle. C-I-R-R-H-O-S-I-S, cirrhosis. And that is a progressive disease that once your liver begins to fail in that way, again, it puts more strain on the surviving cells and you lose more and more until eventually the liver dies. It's technically possible to get cirrhosis from any toxin exposure over time, but quite frankly, alcohol is the only drug that humans regularly and consistently expose themselves to frequently enough and in enough dosage over a long enough period of time for cirrhosis to happen. So it's overwhelmingly caused by alcohol. Not to say that alcohol is inherently harmful, it's just too much over too long. This is the consequence. All right, making some kind of sense? All right, let's move on. Let's talk about a few other particular organelles that you will find in your eukaryotic cell. Let's talk about a thing called the Golgi complex or Golgi apparatus. Either term is acceptable. And if you look at your diagram, you'll see the Golgi complex looks like a little kind of pile of interconnected sacs, little tiny membrane sacs, all connected together. The Golgi complex, and by the way, that word Golgi, capitalize that word. It's a proper name, named after the Italian cell biologist Alessandro Golgi, who first described it. It's spelled G-O-G-L-I, Golgi. No, I just spelled that wrong. I just said Gogli. Never mind. G-O-L-G-I. There we go. Golgi. The whole thing is called the Golgi complex or the Golgi apparatus. You'll see both of those terms used. The individual little membrane sacs within it are called Golgi bodies. In a typical cell in your body, there are 20 or more of those little membrane sacs, Golgi bodies per Golgi complex. You might have more than one Golgi complex per cell. If you're a plant cell, you probably have hundreds of Golgi bodies per Golgi complex. If you're a protist, like a single-celled eukaryote, you probably only have like one or two. Now, the Golgi complex, you can think of it as the packaging plant for the cell. Its job is to gather, package, sort, and distribute various proteins and lipids. It collects molecules that have been produced by other parts of the cell, sorts them out, bundles them up into a packaged form, and then distributes them to where they need to go. What happens is various molecules will enter the Golgi complex. They'll enter through one side of it, an opening called the cis face, C-I-S, that's usually italicized when you see it. And then they'll be sorted by what type of molecule they are into separate individual Golgi bodies. So different Golgi bodies in the same complex can be sorting different kinds of things. Once they've been sorted out in the way, the Golgi bodies will pinch off from the rest of the Golgi complex. They'll exit through the opposite side of it, called the trans face, T-R-A-N-S, often italicized when you see it in writing. Now what you have from that is this little tiny bundle, a little membrane-bound bubble, if you will, that poked off from the Golgi apparatus, got filled up with something, budded away from it, and separated. And now the cell has this little membrane-bound compartments within it. Those little membrane-bound compartments are called vesicles. V-E-S-C-I-C-L-E. Wait, I just spelled that wrong. Sorry. Spelling it out loud, it's different than writing it down. V-E-S-I-C-L-E-S. There we go. Vesicle. Yeah. So vesicles are kind of temporary organelles, if you will, made by the Golgi complex. Each vesicle is a little membrane-bound bubble filled up with whatever we sorted into it. At any given time, your cells have hundreds of vesicles within them. A vesicle is used to move things around within the cell, to transport it from one place to another. They can be used to export things from the cell through a process called exocytosis that we'll talk about in the near future. And they can be used to store stuff, to keep things packed away until such time as the cell needs them. You could have food vesicles. You can have vesicles filled up with enzymes. You can have vesicles filled up with metabolic wastes. A vesicle can basically pack up anything that the cell wants to have packaged and stored away. Now, vesicles sometimes are used to store things that would be harmful to the cell if they were left freely floating around. One type of vesicle that does that you'll find in most of the cells of your body is a type of vesicle called a lysosome. L-Y-S-O-S-O-M-E, lysosome. Remember that prefix lys means to rupture or to burst. A lysosome is a vesicle filled with powerful digestive enzymes. Its internal pH is often as low as 1 or 2. Those enzymes are contained within the vesicle to protect the rest of the cell from them. The vesicle is used when the cell needs to break stuff down. Lysosomes are used for digestive purposes to help cells to break down large complex molecules, like say you have a damaged protein you need to clean up by breaking it down. Lysosomes will do that. They are also used to attack bacteria. In particular, immune cells that have engulfed bacteria and swallowed them up will release lysosomes, which will then attach to the outside surface of the bacterium and then break open, exposing its plasma membrane or its cell wall to dangerous digestive enzymes, which will then cause that cell wall to rupture. And as you guys know, a ruptured cell wall, the cell will swell up and burst. So this is an important part of our body's immune defense, lysosomes. Another similar type of vesicle we find is called a peroxisome, P-E-R-O-X-I-S-O-M-E, peroxisome. Peroxisomes are going to be containing also very caustic enzymes, but their primary job is not in antibacterial effects or digestive effects. Peroxisomes are there for the breakdown of harmful molecules. They collect and break down metabolic waste products that are produced by the cell. In particular, they're used a lot of times in plants in what we call nutrient recovery, meaning that the plant has produced a clunky waste molecule as part of its metabolism. The plants don't eat. They want to recycle as much as they can. So peroxisomes will break down those waste molecules into simple, harmless components that the cell can use again. Animal cells do have peroxisomes as well, but plants tend to have more. All right, so that's Golgi complex, the vesicles that it produces, including lysosomes and peroxisomes. Now, another membrane-bound organelle that you will find in nearly every eukaryotic cell is going to be a structure called a centrosome. C-E-N-T-R-O-S-O-M-E. The centrosome is a structure responsible for producing two kinds of things. One, it's going to produce long protein fibers called microtubules. microtubules. Microtubules literally means little tubes. Microtubules are used to move things around within the cell. They're part of several different cellular structures. And most significantly, the centrosome builds another organelle called the centriole. Centriole. The centriole is an organelle with a number of microtubules extending from it. Centrioles are crucial to the process of cell division. You can't divide a cell without the help of centrioles. We're going to talk more about them when we discuss cell division, but you need centrioles to carry out that complex process. If you don't have centrioles, the cell can't divide. Notable example, neurons, nerve cells do not have centrioles. That's Neuron, neuron. Neurons don't have centrioles, and as a consequence, they can't divide. If you lose a nerve cell, you're not getting it back. And this is one reason why nerve damage tends to be permanent. Like you get your spinal cord cut, you're not going to walk again because your body can't repair the damage from the lost neurons because they can't divide to grow new cells. So the centrioles are important structures that most eukaryotic cells have, but clearly not all. Now, one non-organelle structure that I wanted to say a word about is another structure that contains a lot of microtubules called the cytoskeleton. 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 Remember, prefix cyto means having to do with the cell, so it's the internal skeleton. It's not really a skeleton. What it is think of it kind of like the frame of a building. The cytoskeleton is a framework made out of microtubules and fibers of the motion protein actin, found all throughout the interior of the cell. Some prokaryotes have a cytoskeleton, actually, but they're mostly a eukaryotic thing. And the cytoskeleton is going to provide structural support for the cell, give it shape, give it support. Furthermore, because it contains a lot of motion proteins, like microtubules and actin, the cytoskeleton acts like a rail system. It can be used to move things around from one part of the cell to another, shift molecules and vesicles and things around inside the cell's interior. You will also find that in some cells, the cytoskeleton is used for motility. It can flex and stretch out to allow the cell to extend itself a little bit. In particular, there's a microorganism you might have seen in lab called the amoeba, Amoeba. That's a single-celled eukaryote, a member of the kingdom Protista, a protozoan. Amoebas move by stretching their cytoskeletons out, and that allows them to extend portions of their cell membrane in whatever direction they want to go. The amoeba forms a sort of temporary limb in that way that we call a pseudopod. Pseudopod. Pseudo means false, pod means foot, literally a false foot. And the amoeba reaches out into the environment with these pseudopods, stretching them out from its structure by stretching the cytoskeleton. And that allows the amoeba to pull itself through its environment or to wrap its pseudopods around a particle of food and engulf it. We're going to talk more about how they do that later. So the cytoskeleton isn't really an organelle, but it is an important structure that most eukaryotic cells contain, and some prokaryotes even have one as well. All right, folks, so here's the deal. We are not quite done talking about eukaryotic cells yet. We still need to say a few words about some other structures they can contain. We have not yet talked about specialized things that plant cells have that animal cells don't. And we have not yet talked about some very specialized organelles called chloroplasts and mitochondria, which are used for the purposes of producing cellular energy. So I have a little bit more to say about the structure of eukaryotes. We're not quite done yet. But we'll carry that on into our lecture on Monday when I'm back from my travels. So I want you all, make sure you read this chapter on cell structure if you've not already read it. Read through it, take some notes on it, write down things about it as if you were reading a lecture, and come into class on Monday ready with questions. Questions about the reading, questions about what I just talked about in my lecture here, whether that's a simple question like, can you respell that word for me, although you'll find it in the reading, or whether it's a more complex question like, can you tell me about the differences between smooth ER and rough ER, or can you tell me how cirrhosis works, or any of that kind of stuff. So be ready to ask questions about what you just heard on Monday. I don't have any homework for you. Keep on working on your lab assignments that are due next week, but there's going to be a worksheet in the near future related to cells. But we're not quite done with eukaryotic cells yet, so I won't assign that until later. Probably I'll assign that on Monday. It'll be due to me later on next week. But in the meantime, I have said everything I needed to say to you guys. I hope you're having a great weekend. I'm just about out of time. It's almost exactly 50 minutes here. So I will see you all on Monday. Good night.